If you really enjoyed this lecture, you can watch it a second or third time. Who knows? Tell your friends. And so that's one thing I'll be doing. The other thing I will be doing is I'm going to try and send out some questions to everybody through a device called M-Clickers. So if you have a smartphone or a laptop, now or later on, you can turn them on, get your internet connection working, and then see if I can send you the questions, and then I will get your answers as you put them in, just to see if you've understood something as we go through the talk. I'm going to try a couple of those, okay? So be ready for that. Okay, so my subject today is antiviral immunity. And I'm not going to talk about any one particular virus. So I'm going to give you an overview of the antiviral immune response with examples from different viruses. Now, if you think in a general sense about what is happening during an infection, okay, you'll have, let's say, an uncomplicated, self-resolving acute infection. Okay, you're infected at day 0. You will have a peak of virus replication. Then first, you're going to have the innate immune response, which is activated. So you'll have interferon and other acute phase cytokines. You'll have fever, you'll feel bad. And this innate response will be accompanied or followed by activation of NK cells, natural killer lymphocytes. And then slightly later, the adaptive immune response will kick in with first the appearance of CTL, cytotoxic T lymphocytes, and then seroconversion with the appearance of specific antibodies against the virus. Okay, so this interferon part and the NK part, that's the innate response. CTL and antibodies, they are parts of the adaptive response. Now it's very important that all of these things actually happen during an immune response, because if it doesn't, then your pathology is going to be more severe. So for example, people who have got an Ebola virus infection, the people who actually can survive, they have symptoms and they survive, they have a lot of activated CD8 positive lymphocytes. They have a lot of CTL activation. People who die from their infection, they do not have CTL activation, and they have 1,000-fold higher viral load. So in that case, this part of the response is lacking, and the consequence is very severe. Okay, so that's generally what happens in a viral infection. So what I'm going to talk about today is, first we're going to look at early events during infection. What happens when viruses infect cells in the body? How do cells realize that there is a viral infection, and what happens? Then I'm going to talk about NK and CTL together as part of the cellular response, and then antibodies. I'm not going to talk about CD4 helper cells or cytokine networks. These are important, of course, in order to orchestrate the adaptive response. But my focus today is on antiviral immune effectors, okay, the effector mechanisms that really limit viral replication. So that's why I'm missing out CD4 helper cells and cytokines mostly. But okay, don't imagine that these things are not important. Okay, so for the first question, whenever you have an infection is, how does a cell know that it's infected? And this is not such a trivial question, in fact, because there's no big biochemical difference between virus particles and any other protein floating around in your body. It's not like bacteria, which have got LPS or peptidoglycan that is biochemically very distinct. So it's not entirely obvious how cells in your body can differentiate a virus particle from something else. And in fact, what seems to be the main, the main trigger to start up the innate immune response is the detection of viral nucleic acids. I'm not going to talk too much about this because I think Jacques Lecerf next week will be telling you a bit more about these receptors and how they induce interferon. But I just want to say two things, okay? So there are two types of these receptors. It's the toll-like receptors, which are expressed by dendritic cell subsets. And when these get activated by RNase or DNA, then it's going to induce maturation of the dendritic cells, and that's going to be essential for the initiation of the adaptive response. Okay, so if you don't have DC maturation at some stage, you're not going to have any antibodies, you're not going to have any CTL. The other family or large class of nucleic acid receptors that exist are cytoplasmic. So these are going to detect viruses or nucleic acids that viruses produce inside the infected cell. And the main result of this is that infected cells are going to produce interferon. Okay, so you might ask yourself, okay, how come these receptors can actually distinguish viral nucleic acids from cellular nucleic acids? We've got loads of RNA and DNA inside cells. Generally speaking, I think you can say that these receptors are detecting either nucleic acid in the wrong place. Okay, if you've got DNA inside the cytoplasm, that is not normal. Okay, could indicate viral infection. Or they are detecting biochemical forms of RNA that are not normally produced inside cells. So cells don't normally have a lot of double-stranded RNA, but that's produced as an intermediate in RNA virus replication. Also in the cytoplasm of cells, you don't really have RNA with a 5' triphosphate at one end of it. Okay, messenger RNA, it's got a 5' cap, right? Doesn't have a free phosphate at the end of it. So this RIG1 receptor is recognized as something that is very, very specific, and you only find in viral RNAs. Okay, so that's how your cells find out or realize that they are being infected by something. Okay, they detect viral nucleic acids. And that's the answer to our first question. The second question is, okay, you're infected. What do you do if you're infected? There are two things that cells do. First, they secrete interferon. So most cell types in the body, if they're infected with a virus, they can secrete interferon beta or beta interferon. Now this is an antiviral molecule. It doesn't have any direct effect on viruses themselves, but they induce an antiviral state in the surrounding cells. So all of the cells in the tissue that bind interferon beta through the interferon type 1 receptors are going to become much more resistant to viral infection. And that's the first thing that happens. The second thing that happens is the infected cell itself is going to die. And that's important because if an infected cell can kill itself before virus replication is completed, then that will limit viral spread in the tissue and in the organ. So programmed cell death after viral infection is a very important part of our intrinsic defenses against viral infection. So that's generally what the response is at a cellular level against viruses. Now what I'm going to talk about next is exactly how this happens. Okay. Right. So interferon. Once you've said it induces an antiviral or a virus-resistant state in cells, you understand the concept. But in terms of mechanism, what is that exactly is happening? Well, what occurs is that once cells bind interferon with the interferon type 1 receptor, then they will start transcribing interferon-sensitive genes or interferon-induced genes. And many of these interferon-induced genes are restriction factors for viruses. That is, they are cellular proteins that are going to block one or other of the different steps in the virus replication cycle. Okay, so they may be constitutively expressed at a low level in many cell types, but as a general feature, their expression is greatly increased after by interferon treatment. Now you might wonder, why, if we've got all this fantastic antiviral machinery, how we ever get infected. In fact, most viruses that infect human beings have evolved mechanisms to evade these antiviral restriction factors. So most of these restriction factors can somehow be undermined by what we call virus accessory genes. Okay, so in a virus genome, it's easy to identify three types of genes. Structural genes, they're involved in the capsid, in making the virus particle. Genes involved in virus replication, might be a polymerase, for example. And then once you've identified those genes, you're left with a whole bunch of other virus genes, which for a long time people didn't really know what the function was. So these are what you call accessory genes. And it turns out that most of these, or many of these accessory genes, their function is to try and get round the defenses of the host cell, to make sure that the virus can actually succeed in infecting us. And then what tends to occur is that both the viral accessory genes and the host restriction factors are under selection pressure. And this leads to host virus adaptation and it's important in the species barrier between, that prevents us from catching viruses from a whole bunch of animals. Okay, now what I'm going to do, I think Mr. Lasik is going to talk a little bit about ADAR1 and maybe RNA-Zel next week. So I'm going to talk about all these other intrinsic antiviral restriction factors so that you understand a little bit about how they work. Okay. But first, how do we actually know these things exist? How did anybody ever, identify them? Well, one of the approaches that's been used successfully to identify many restriction factors is to try and consider what is happening if you've got a virus and two cell lines from the host species. One of them is permissive for the infection, so the virus can replicate effectively in the permissive cell line. This other cell line B here, it's non-permissive. So the virus can maybe bind and enter, but it can't actually replicate effectively in cell line B. Once you have that situation, there are two possible explanations. Either B, okay, this cell line here, it's lacking some cofactor that is essential for virus replication. Or it's expressing a restriction factor that prevents the virus from replicating. Now the way to choose between these two options is to fuse these two cell types and produce a heterokaryon and then try and infect it. So if you have a restriction factor, then this fused heterokaryon will be non-permissive. Okay, so restriction is dominant. Okay, if this cell was lacking the cofactor, then making the fusion with the cell A, that should bring the cofactor. So this should be permissive, right? If it's you're talking about a cofactor. So in many cases, this was the first step to identifying restriction factors. You get two cell lines or two cell types, you try and fuse them. If the heterokaryon is resistant, still non-permissive, then there will be a restriction factor. And then you can try and clone it because you have this cell type expresses the restriction factor. This cell type doesn't express it. And you can try and make cDNA banks from this cell, transfer them into cell type A and see which cDNA clone will make this cell type non-permissive. And that cDNA clone will code for your restriction factor. So this is the general approach that was taken to identify these things. Well, not every time, in fact. Okay, so that's how they were identified. So let's go and have a look at different restriction factors and how they act. TRIM5 alpha. Okay, this was identified because it was noticed that rhesus macaque cells are not permissive for infection by HIV-1. So we had permissive cell, non-permissive cell, and it was possible to clone the macaque gene that was responsible for this restriction. It turned out to be TRIM5 alpha. And it has this kind of structure here. It's got a ring domain, coil domain. This causes a dimerization. And then this pri-spri domain, which is the part of the molecule that binds directly to the retroviral capsid. So what happens is as soon as this capsid enters the infected cell, TRIM5 alpha will bind to it, and it's going to prevent decapsidation. So it stops the viral nucleic acid from getting out, and it stops the replication at a very early step. Now the effectiveness of TRIM5 alpha is dependent on this interaction, direct interaction between TRIM5 alpha, the pri-spri domain, and the capsid protein. So of course, retroviruses escape. They mutate their capsids, and they escape from restriction by TRIM5 alpha. So that's why, human TRIM5 alpha does not prevent HIV-1 infection, because these variable loops that are involved in binding the capsid, they don't work on HIV-1. So that's why we can be infected by this virus. Well, okay, one of the reasons. But, you'll be happy to know that we can't be infected by mouse retroviruses because TRIM5 alpha is keeping us safe. Okay? The next one is a molecule called IFI-TM, which is for interferon-induced transmembrane protein. Now this one was identified kind of by accident because there was a group looking for cofactors necessary for influenza virus infection. So their idea was that they would have a large-scale screening, knock down all the genes in the cell one by one, and then infect those cells with influenza virus. When influenza couldn't replicate, that meant that the gene they knocked down was a cofactor. Except that they found one gene, when they knocked it down, influenza virus replication was increased. So they thought, okay, this is a restriction factor. It's present. When it's present, it limits influenza replication. So then they went after this one and said, okay, what is the function of this molecule? So they identified IFI-TM3, and then realized that all the members of this family, IFI-TM1, 2, and 3, have a similar structure and function. And their role is to inhibit the entry of envelope viruses. So IFI-TM1 is mostly found in the plasma membrane or early endosomes, and it blocks the entrance of viruses that enter either directly at the plasma membrane or soon after internalization. So IFI-TM1 can block HIV or hepatitis C entry. IFI-TM2 and 3, they're expressed in late endosomes, and they can block the entry of viruses that require a very acidic environment to induce membrane fusion. So IFI-TM2 and 3 are effective against influenza and dengue fever viruses. Okay, so how does this work? How does this thing work, right? So we really don't know exactly what the precise mechanism is. Okay, this was discovered in 2009, quite recent. Now the name of this molecule is IFI-TM, TM for transmembrane. And that was just because by looking at the sequence of the protein, there were two hydrophobic domains. So initially it was thought that these two hydrophobic domains crossed the membrane somehow. But currently it seems that these two hydrophobic domains just sit on the cytoplasmic side of the membrane. So maybe transmembrane here is not exactly correct. So how is it working, all right? Well, there are these three different models. Firstly, okay, for envelope viruses to infect the cell, often they need to, the attachment glycoprotein needs to interact first with a cellular receptor and then with a co-receptor. And it's this glycoprotein receptor co-receptor complex that's important. So one idea is that IFI-TM will kind of rigidify the host cell membrane and prevent lateral diffusion, so that once you've got the attachment of the virus to its receptor, the co-receptor is kept at a distance. So this essential complex can't be formed. Something quite similar to this idea is that okay, maybe you do get interaction between the attachment glycoprotein, the receptor, and the host cell co-receptor, but you just get one of them. For fusion to occur, you need lots of these complexes to come together at a kind of synapse between the virus particle and the cell. So maybe once again, IFITM will limit lateral diffusion and prevent these complexes from associating. And finally, the other model is that perhaps it doesn't do any of this at all. Maybe viruses do form a kind of pre-fusion complex, but it's just that IFITM will modify the physicochemical properties of the host cell membrane, and it's going to prevent the formation of this fusion pore. Okay, this is the essential event for entry of envelope viruses. On the blue side here, this is the virus side, this is the cell side. Once you've got this pore here, everything that's inside the virus particle can enter the cell. So if IFITM can prevent this pore from opening, it's going to prevent infection. Yeah, so we have two restriction factors that work at very, very early stages of virus infection. ApoBec. Okay, this one's going to be a little bit more complicated. I'm sorry to announce that, but it is. Okay. So there are a whole family of ApoBec proteins. They are restriction factors. And they were discovered because it was noticed that some cell lines were permissive for HIV-1, which was deleted from one of its accessory genes, the gene VIF, whereas other cell lines were non-permissive for these delta VIF viruses. And then once again, you've got the permissive cell line, non-permissive cell line. You can try and clone the restriction factor. And this was found to be the gene ApoBec3G, which confers this non-permissive phenotype. ApoBec3G, how does it work? Well, the reason it's complicated is that a cell which expresses ApoBec3G can be infected by virus, the HIV. Okay? But in the absence of VIF, this cell that's infected will release virus particles that have got ApoBec3G incorporated into them. And these virus particles will be non-infectious for the next cell that's going to be infected. Okay, so ApoBec3G isn't actually preventing infection in the first cell, but it prevents transmission of an infectious virus. So how does that work? Okay, and the role of the HIV accessory protein VIF is to prevent ApoBec3G from being incorporated in the virus particles, and that allows infected cells to release infectious particles. Okay, how does it work? ApoBec3G is a cytidine deaminase. Its substrate is cytidine in DNA molecules. And once HIV enters the infected cell, first you have decapsidation, then you have reverse transcription. So the HIV reverse transcriptase copies the RNA genome into DNA. While this is happening, ApoBec is already inside the particle. So as soon as the virus starts copying its genome into DNA, ApoBec starts mutating it. So what it's going to do is it's going to get hold of these cytidines in the first strand of cDNA that's synthesized by the reverse transcriptase, and it's going to cleave off an amine group. Once you cleave off that amine group, you've got uridine. Okay, so this is on the minus end strand. So the net effect is that you're going to have a mutation of G to A in the plus end strand. So that's the net effect of ApoBec. It's going to induce G to A hypermutation in the virus genome. That's going to be very harmful for the virus because, well, one of the things is that there is a sequence specificity. It doesn't just pick on any C to deaminate. It's got sequence specificity for this CCA kind of group here. And if you look at that, then it's part of the codon for tryptophan. Because it's going to be this TAG in the DNA is going to be mutated to, TGG is going to be mutated to TAG. So when this protein is getting translated, tryptophan is going to be converted to a stop codon. So a lot of stop codons are going to be introduced into HIV protein. And that's it. Your virus is dead if that happens. Because once this genome gets integrated, starts being expressed, you can't get functional proteins. So that's very clever, and it's very useful to protect us against retroviruses. SAMHD1, this was the last one to be discovered, I think, for HIV restriction factors. Once again, it's looking at these accessory proteins in HIV and associated retroviruses. So in fact, HIV2 has got an accessory protein called VPX, which is absent in HIV1. And what's also true is that HIV2 can infect macrophages efficiently and HIV1 can't. Now, in fact, this is due to the VPX protein, because when you delete VPX in the HIV2 genome, they can't replicate in macrophages either. So this was what was known as the myeloid block to HIV1 replication. So several groups were looking at, okay, what is the protein responsible for the myeloid block? And this time they used a biochemical approach. They looked for proteins that bound to VPX in the macrophages, and they pulled out this one, which is called SAMHD1. Now it turns out that SAMHD1 is a kind of nuclease, and its substrate is deoxynucleotide triphosphates. Once SAMHD1 gets activated, it will degrade the DNTPs into deoxynucleotides. DNTPs, you need them to synthesize DNA, right? So a retrovirus comes in with its genome. It's got a reverse transcriptase. It's got to synthesize DNA. It needs DNTPs. So this works against retroviruses and DNA viruses because it starves them of DNTPs and prevents them from replicating their genomes. Or at least that was the model in 2012 when this was just shortly after this molecule was discovered. But more recently, some people have said, no, it's not that. SAMHD1 has an RNase activity and it destroys HIV genome directly. So this one is not entirely sure how it works yet. There's a little bit of controversy. Okay, we have one more restriction factor. Everyone can just keep alive till we get to the end of restriction factors, and then we're going to go on to something else. The last one is tethering. And once again, it's looking at these accessory proteins in HIV1. This time it's a small protein VPU. When this protein is deleted, it was found that HIV1 cannot liberate infectious particles very effectively, and they remain attached to the surface of the infected cell. And it was found that this was due to a restriction factor, which is called BST2 tethering. Okay, when you look at the structure of this thing, it's got a very unusual structure. It's a protein that's anchored in on the outside of the cell membrane at its C terminus by a GPI anchor. And it's also anchored by its N-terminal domain with a transmembrane domain. Now, retroviruses and many viruses bud through regions that are rich in GPI anchored proteins. So this part here is going to be directed to the site of HIV1 budding, whereas this part is going to remain stuck in the cell membrane. And the result is that when these particles are released by budding, they can't get away because they have a GPI part, which is in the viral membrane here. And they have this alpha helical transmembrane domain that is holding the cells, holding the virus particles onto the surface of the cell. So you don't get effective release of virus particles, and they all stay stuck on the infected cell. That's very good as an antiviral mechanism because it means that the infection is not going to spread in the body. Okay, and this is a general antiviral mechanism because it turns out that this molecule is involved in resistance against filoviridae like Ebola virus, Marburg virus, paramyxoviridae like measles, this kind of thing, rabies virus, and herpes viruses. All these generally speaking, all these envelope viruses are sensitive to tethering. But they can still infect us. What does that mean? It means that they have some kind of protein that is inactivating tethering. So for HIV-1, it's the VPU protein, which binds to tethering and needs it to be degraded. In HIV-2, HIV-2 doesn't have VPU, but it's the envelope glycoprotein that will bind to tethering and lead it to be degraded. SIV, the simian immunodeficiency virus, it's the NEF protein, which leads to degradation of tethering. And for Ebola virus, it's the surface glycoprotein, which will inactivate tethering. So all these successful viruses that can infect us, they've found a way to avoid being held onto the surface of the infected cell by tethering. Okay, so this leads me to the last point on restriction factors, and that is that the restriction factors are an important part of host virus coevolution. So one part of it is that, viruses are dependent on the cofactors of the host. So for example, the cell receptors might be variable in a population, and the virus has got to adapt to that. But the other half of this host virus coevolution is that viruses have always got to adapt to host restriction factors. And this is just kind of like illustrating that. So you'll have a host cell restriction factor here, and some kind of virus protein has got to target it and get it degraded in order for this virus to be successful. And this is going to induce a selection pressure at the level of the host population, not individual cells or even individual people, but in the whole population. There will be people who have got a polymorphism in the genes coding for these restriction factors. And the people who survive infections better are going to be the ones who express a mutant form. So the host cell population is going to change, and that's going to induce selection pressure on the virus. So the virus has got to mutate its antagonist so that it can once again degrade this host restriction factor. And then this whole thing goes on and on and on and on in a big circle. So host cell restriction factors and viral antagonists, they're continually adapting to each other. So that means that the viruses become very, very specialized at infecting one particular host. And that's why, generally speaking, we can't catch cow viruses or sheep viruses, this kind of stuff. Even though people in agriculture, farmers are getting in contact with them all the time. So that explains, to a large extent, why zoonoses, viral infections from animals, are relatively rare events. Not impossible, but not very frequent. Okay, so that's the first part. Infected cells secrete interferon. Interferon will induce restriction factors in surrounding cells, and these cells become much more difficult for viruses to infect. The next part of the second part of the intrinsic cellular defense is infected cells die. Now, often you might think, okay, no wonder this cell dies. It's infected by a virus, you idiot. Of course it's going to die. But that's kind of looking at things in the wrong way, because viruses don't really want to kill cells. They just want cells to become virus factories. And if the cell can stay alive for a long time, that's good for viruses. Often, the death of virus-infected cells it's a cellular response to viral infection. Okay? And this can occur through three pathways. Apoptosis, which probably everyone will be familiar with. And the main event here is activation of caspases, either through the activation of death receptors, through the Fad caspase 8 pathway, or through the mitochondrial pathway. And if these caspase 3, caspase 7 are activated, then you get apoptosis. This is non-inflammatory cell death. That's one mechanism of programmed cell death. Another mechanism is pyroptosis. So in this case, you get activation of caspase 1 by this kind of ASC, adapter protein adapter complex. So caspase 1 will cleave IL-1 beta. So it's going to be pro-inflammatory. It's going to cause the death of the cell, but cause inflammation at the same time. Okay, that's why it's called pyroptosis. It's pyretic. And somewhere in between the two, there's this weird thing called necroptosis. And necroptosis, the main event is activation of these kinases, RIP1 and RIP3. And if they are activated, then you'll get a membrane rupture, so it's a kind of necrosis, but without inflammation. Now, necroptosis seems to be like a fallback mechanism because cells can die by necroptosis even when caspase 8 is inhibited. So it's almost like, if you're going to, the first attempt for a cell to commit suicide is going to try and induce apoptosis. And if this fails, then the necroptosis pathway will be activated. So it's kind of backup, okay? Now, viruses don't like their host cells to, well, before I get to that, so I'm skipping a slide. Viral infection induces cell death. How? Okay, what is it that induces cell death in infected cells? Once again, it turns out it's mostly detection of viral nucleic acids that will induce apoptosis or other forms of cell death. If you have double-stranded RNA detected inside an infected cell, it will activate RNA-dependent protein kinase, and this can activate FAD and caspase 8 and activate apoptosis. Other RNA receptors are involved in activation of the ASC caspase 1. That's pyroptosis, right? Yeah. Caspase 1, IL-1 beta, inflammation. The same thing can happen with the presence of double-stranded DNA. It can be detected by these receptors, and they activate pyroptosis. Necroptosis seems to involve activation of this DNA receptor, and the mitochondrial pathway of apoptosis seems to be activated during viral infections by activation of p53. There's kind of detection of DNA damage. Normally, you would think about this in cancer, but it's also involved in viral infection. And these kind of processes can be very, very important in pathology. Okay? In HIV infection, it's been known for a long time that a lot of CD4 T cells die in the periphery, and the vast majority of these cells are not productively infected. What is happening is that viruses entering these cells, HIV DNA after reverse transcription is detected by IFI16, and it induces pyroptosis. So this is connected to death of CD4 T cells and kind of chronic immune activation due to this the activation of this pyroptosis pathway. So this is something that we've understood just like in the last year. Okay, so cells try to die. They do their best to die after they get infected. But of course, viruses don't like their host cells to die. They want them to survive, at least until they can produce more viruses. So lots of viruses have accessory proteins that inhibit different steps in the apoptosis pathway or other pathways of cell death. So many viruses will inhibit caspase 8. So the adenovirus, pox virus, cytomegalovirus. Many different viruses will inhibit the mitochondrial pathway of cell death, of apoptosis. This is Epstein-Barr virus. This is cytomegalovirus, adenovirus again. And pox viruses can inhibit the effector caspases. The other pathways of cell death, necroptosis and pyroptosis, have been less studied. Okay? So we only know one viral protein that can inhibit necroptosis. But that doesn't mean that's the only one. It just means that they haven't been studied so much. Okay? We know about apoptosis for quite a long time, but these necroptosis and pyroptosis are relatively new. So I don't think people really have identified all the viral proteins that can prevent cell death by these pathways. Okay, now it's time for a little break, right? So what I would like you to do is, if you have a smartphone or a laptop, get your internet connection going up, and connect onto this URL. Okay. When you're ready, I'll send out the question, then everybody can think about it and send in their answers. Okay? Okay, let's see if I can. Have you got it? Oh, you want the address again? Sorry. Okay, it's here. You're going to be connected onto mClickers. Actually, I need to put the session key in, okay? So let me just start. Okay, it's down here. I'll put 68679. Okay, that's the session key. Okay, everybody got it? So if you've done it, you should get some questions coming up. Oh, let me show. It's this. Can you see that? You got it? Okay. So once you have it, think about these questions. Try and answer them. It's which one is true? Maybe one, maybe several, maybe zero. Okay? So I just want everybody to answer and see if you've understood something about what I've been telling you. No responses submitted so far. Oh yeah, I'm getting some answers up here. Oh, don't look. I'm going to hide the results until everybody sends something in, okay? Is that okay? And when everybody sent some answers? Some people still thinking about it? I've got 8 responses so far. Must be a few more, right? Okay, let's see what you think, okay? Shall we have a look? So, oh, response is 9. Where's the graph? Oh, supposed to show me the graph here, right? Okay, right, so answer one. Which of these are true, right? Restriction is a dominant phenotype. 7 people said yes. You're right. That's it. Restriction is dominant. The heterokaryon should have restriction. Restriction factors are strongly expressed in a constitutive fashion. 2 people said yes. You are wrong. Okay, you fell into the trap. They can be constitutively expressed, but at low level. Okay, strong expression is after interferon. Next one. Each restriction factor can target multiple viruses. That's true. Okay, 6 people, you are right. Okay. Generally speaking, that's true. They don't just target one virus. They might just target one particular family of viruses. You might have retroviral specific restriction factors. But it's not just one virus. There will be many members of this virus family, or there will be more general than that. And the opposite is also true. Okay, a particular virus can be targeted by multiple restriction factors. Okay, that's good. Everybody got that one. And then finally, RNA viruses are more sensitive to restriction factors than DNA viruses. That's wrong. Okay, so everybody got the correct answer here. Okay, good. Okay. That's nice. I'll try and give you another question a bit later on, okay? See how we do for, depends if we have time. Okay, so that's all right. Everybody's doing well here. Right, so the session key will be the same, all right? So you don't need to change anything when we get to the next question. And now we're going to look about the adaptive response. Okay, well, adaptive NK cell and CTLs and then antibodies. All right, so what is the purpose of the NK and CTL response? Well, if a virus-infected cell here is not going to kill itself, all right, it's tried to enter into apoptosis, but the viral proteins are blocking cell death. CTL and NK cells can come along, recognize the virus-infected cell, and that's it, they'll kill it. Okay? So they are the liquidators. Once again, the main question is, how do you recognize that this cell has got a virus infection? For NK cells, maybe not so obvious, right? So here is my simplified understanding of how NK cells identify virus-infected cells. It has to be simple because that's about all I can understand as far as NK cells go. So natural killer cells, they express a variety of inhibitory receptors, like the KIR receptors, and some activating receptors, like NKG2D. And whether or not this NK cell gets activated depends on the balance of signals it gets from the inhibitory receptors and the activating receptors. So when an NK cell encounters a non-infected cell, the KIR receptors will interact with the HLA class 1 molecules on the surface of this cell. The NK cell will get an inhibitory signal and it will not kill this cell. Okay, what happens during virus infection? This cell starts accumulating viral nucleic acids, virus proteins. One of the things that happens is that HLA class 1 expression on the surface will be reduced. So the inhibitory signal is going to get lower, get smaller. And additionally, virus infection leads to the expression of these ligands for the activator receptor, like MIC-A and MIC-B, and ULBP 1 to 6. So the net result is that this NK cell, when it meets a virus-infected cell, is that the inhibitory signal is very weak, and there is an activating signal. And this is why, and that leads to expression or secretion of cytotoxic effectors into the virus-infected cell, which will be killed. So don't forget that NK cells, they're part of the innate response and they're activated and their proliferation is induced by type 1 interferon. So to get a very active NK cell response, you need to have interferon. Now, of course, viruses want to try and avoid being killed by NK cells, and in particular, herpes viruses. These viruses seem to be very, very sensitive to the NK cell response. So cytomegalovirus, human CMV and mouse CMV, have a number of proteins that inhibit the NK cell response. So UL16, this is a CMV protein, going to prevent these activating ligands from being expressed by the infected cell. And this one as well, UL142, will be retaining MIC-A. So it's going to knock down the activator signal. And secondly, CMV infected cells express UL18. It's a homolog, not a homologue, homolog of HLA class 1. So that means that you have UL18 here, which is going to interact with the KIR, so that these NK cells will get an inhibitory signal. So that's how one particular virus evades the NK cell response. So that's it for NK cells as far as I'm concerned. What about the CTL response? Okay, so as you probably know, CTL recognize viral peptides that are presented on the HLA class 1 molecules on the surface of an infected cell. So these virus peptides get there because when viral proteins are expressed, they can be sampled by the proteasome that produces peptides. They get transported into the endoplasmic reticulum, loaded onto HLA class 1, and then that goes to the surface. Now, what is perhaps less well known is that in some cases, virus capsids, as soon as they enter the cytoplasm, can be disassembled by the proteasome. So even before you start to get expression of viral genes, virus proteins that enter the cell can be degraded and then presented on HLA class 1. Now, the point of CTL and NK cell lysis of infected cells is to prevent these infected cells from releasing virus particles. And the quicker you can lyse an infected cell, the more effective your antiviral CTL is going to be. So that means that the consequence of that is that CTL that are effective, that have a good antiviral effect, are the ones that target either early genes, first genes that the viruses are going to be expressing, or they target capsid proteins that enter the virus cell, enter the infected cell. Okay? So that these cells can be lysed very, very, very quickly after infection. No point in like recognizing a very late protein, because this cell is probably going to be full of an infectious virus anyway. Okay, so that's kind of the basis of what makes an effective CTL response. So if you look at the CTL response against cytomegalovirus, most of the response is going to be targeting these either proteins that are in the tegument, they come into the cytoplasm just after envelope fusion, or immediate early proteins. And in the case of HIV, it's the GAG-specific response which is the most efficient, because you can lyse cells even before they start expressing viral proteins. Now, as you might expect, viruses try and escape from CTL lysis, and there are two main mechanisms. For viruses which have a very inefficient, well, not inefficient, but not very high fidelity polymerase, like RNA viruses or retroviruses, they induce a lot of spontaneous mutations in every round of viral replication. So these viruses tend to escape from CTL responses just because they generate a lot of mutants, and the mutants that escape from the CTL response are selected. So how do these mutants render the virus resistant to CTL? Well, some mutations within an epitope can modify processing of the protein by the proteasome. So if your epitope, like this SL9 one here, is cut in the middle by the proteasome, it can't be presented anymore. So mutations that occur that modify proteasome processing will change the epitopes that are on the surface of the infected cell. Some mutations outside the epitope on the N-terminal, or rather, the N-terminal side of the epitope, can modify the trimming step that occurs in the endoplasmic reticulum. And if you can't trim down this precursor peptide, then it can't be loaded effectively into the class 1. Other mutations within the epitope can modify the amino acid side chains that are essential for binding to HLA binding. So these peptides are produced, but they can't stick to HLA class 1 anymore. So they're no good as epitopes. And finally, even if your epitope is processed and presented on class 1, maybe it's the amino acids that are directly contacting the T cell receptor that are mutated. So the CTL doesn't recognize this mutant epitope anymore. So these are different mechanisms by which mutation can lead to escape from CTL responses. Okay, it doesn't all go in the virus's favor, because some of these mutations might touch essential proteins for the virus, and they'll be associated with a fitness cost. So that is to say, if you isolate the virus, so a good example is the response to HIV-1. This capsid epitope is restricted by HLA-B27. People who are infected with HIV-1 and who have this HLA allele, they have a slower disease progression than everyone else. That's because their CTL response is really great. And the reason it's really great is the virus can escape from this CTL response. But if you isolate the virus that has the escape mutation, you notice that in fact it doesn't replicate very well. And that's because this part of the capsid protein is is essential for the virus. If it mutates, then the virus is not so good. So if the virus doesn't incorporate the escape mutation, all the infected cells get killed by the CTL and the viral load is lower. But if the virus does escape, it doesn't replicate so well and the viral load is still lower. So that's why people with this HLA allele do better. That is in the absence of treatment. Okay. So that means in a general sense, okay, so the CTL responses that are going to be effective are the ones that are targeting proteins that are essential for the virus, or epitopes that are very difficult for the virus to mutate. One counterexample is a CTL response against HIV envelope. It's useless. There's very, very little selection pressure to keep these epitopes the same. So the virus can mutate and escape from the CTL response, and it replicates just with no problem. There is no fitness cost. So that's the wrong target to go for, in fact, for CTL. So the other way that viruses can escape from the CTL response is to express proteins that inhibit some part of the processing and presentation pathway. And this is what happens in many DNA viruses. Again, it's all these herpes viruses, okay? So they're DNA viruses, their DNA polymerase has a higher fidelity than reverse transcriptase or RNA polymerases, so they don't produce so many mutants. So they need another way to escape from CTL. So some viral proteins can inhibit proteasomal processing. Some can inhibit the transport of the peptides into the endoplasmic reticulum. So herpes simplex virus 1 expresses this protein. It binds to the cytoplasmic face of the TAP complex and prevents the peptides from getting imported into the ER. The US6 protein of cytomegalovirus does the same thing, except it binds to the luminal face of the TAP complex. Other CMV proteins get hold of MHC class 1 complexes in the endoplasmic reticulum and cause them to be degraded. And HIV-1 NEF can also cause the internalization of MHC class 1 from the surface and cause it to be re-internalized and degraded. So of course, when this happens, these cells will express less class 1 on the surface, and there'll be a target for NK cells. Okay, so that's why NK cells are important, because viruses are always trying to escape from CTL. And if they do that, a lot of the time they will reduce the HLA class 1 expression on the surface. And that makes these infected cells susceptible to NK cell lysis. That's why you need both of these effector systems. Okay, so I was saying we need them, but what exactly is the role of these things in an infection? Okay, we considered these things from a kind of cell point of view. What about a whole organism point of view? So in many models, animal models of acute infection, you'll find that NK cells are activated and present before CTL, part of the innate response. Then you get the CTL after about a week or so. And presence of NK cells and CTL often correlates with the reduction in the peak viral load. And then after a couple of weeks, you'll get seroconversion. Now, it's been shown in a lot of animal models, if you eliminate CTL, then infection is much more severe. So that's the role in vivo of the CTL responses, controlling viral infection in the first week or 2 weeks after the infection event. That's to say, in an acute infection, okay? Now, in persistent infections, for example, latent infections, now once again, it's these herpes viruses, they all produce latent infections, then CTL are always present and they are important in preventing reactivation. And we know that because if people are immunosuppressed, for example, they've had some kind of transplant and they are under immunosuppressive medication, then all of these herpes viruses can be reactivated and cause problems because there's no more CTL that are controlling them anymore. Now, in chronic infections like hepatitis C and HIV, CTL, once again, they're active throughout the chronic infection, and their role is to limit viral replication. At least partially. They never manage to eliminate the virus, but at least they're preventing the virus from completely taking over the whole of your body. Although I have to say that, in fact, maybe 15% of people who get infected by hepatitis C will in fact succeed in eliminating the virus. And that is, well, pretty much due to the quality of their CTL response. Once again, you see people with a particular HLA class 1 alleles can eliminate hepatitis C more readily. And if you don't have the right HLA class 1, you're not responding to the right virus epitopes, and you'll get a chronic infection because your CTL response is going to be directed against epitopes that the virus mutates quite easily. So in these situations, there's a kind of, it's like the First World War, okay? The front lines, they don't move, but you have like millions of cells being killed every day. And you're always having a kind of a race between the viruses mutating, the CTL are trying to catch up. And the result is a kind of stalemate, or at least some kind of equilibrium. So that's the role of CTL in natural infection. But because they have an antiviral effect, that's pretty clear. Can you harness this antiviral effect to prevent subsequent infections? That is, do they play an important part of your memory response? So in mouse models, okay, then one of the things that was done classically was you've got an infected mouse, it develops a CTL response. Can you transfer protection if you transfer the cell? The answer is yes, in many different viruses. So for influenza, West Nile virus, or even Ebola virus, if you transfer CTL then into a naive mouse, then infect the mouse, it can be protected against pathology. Okay, so CTL can be transferred, it transfers protection. Can you induce protective CTL by vaccination to prevent infection? So once again in the mouse, yes, it works. So this is mouse cytomegalovirus, and most of the CTL response is directed against this one protein, protein 89. And if you vaccinate with this protein, then it can protect the mouse against the whole virus. And that's due to CTL. So vaccination to induce CTL can prevent subsequent virus infection in animal models. This is the important message here. Does it work in human beings? Now, the only data we really have on this comes from a large-scale phase 2b trial of a candidate HIV vaccine, which was produced by Merck. And the candidate vaccine was a recombinant adenovirus that expressed GAG-POL and NEF proteins. And the objective of the vaccine trial was to induce CTL, that were HIV specific. And in that sense, it was a success, okay? 73% of the people vaccinated developed HIV-specific CTL against at least one of these targets. Second objective was to prevent infection or reduce the severity of the pathology. How about this? Okay, this was a randomized placebo-controlled trial, double-blind. When the results were unblinded, it was found that there were 21 infections in the placebo group and 24 infections in the vaccine group. So there was no evidence of protection, even though the vaccine had been able to induce CTL. Okay? So this is a major message here, right? So far, CTL have an antiviral effect, have an important role in the natural progression of acute and chronic viral infections. But so far, they can't be harnessed by vaccination. Okay? Vaccines don't work, or at least the vaccines that we currently have, do not actually work by inducing CTL. Okay, before we go on to the humoral response, shall we do another question? Or do we want to keep on going? Maybe we should keep on going because you can show me, you could put hands up if you like. Who wants another question? Okay, nobody, right? Okay, we keep on going. So, let's keep on going. We'll go to do the humoral response. If we have time, we'll have a question at the end, okay? Humoral response, antibodies. Now, I'm not going to talk about antibodies that bind onto infected cells and introduce antibody-dependent cell cytotoxicity. My main focus here is going to be on neutralizing antibodies. These are antibodies that can directly block infection by either binding to the virus glycoprotein or attachment glycoprotein or attachment capsid proteins. And this will block the interaction between the virus and its receptor or its co-receptor. Or in the case of envelope viruses, some types of neutralizing antibodies can block the activation of the fusion domains that are necessary for viral entry. Okay, so it looks like this. This is a rhinovirus particle which has been complexed with the extracellular domain of its receptor, ICAM1. So you can see that the receptor is binding to this region of the capsid. So this is the same thing except with a neutralizing antibody. So the neutralizing antibody binding onto the capsid at exactly the same sites that are involved in attachment to the receptor. So once you have your virus particle covered with antibody, there's no way this thing can bind to its receptor and it can't attach to a host cell. So it's going to be non-infectious. Now the reason antibodies are very important in long-term protection is because they can be present, secreted at the mucosal surfaces. Okay, so they can be in your genital mucosa, they can be in your saliva, and they can be there to prevent a virus particle that enters your body, and it can prevent even the first cell from getting infected, right? And of course, it's long-lasting. You can be seropositive. You have antibodies that can last for years or decades, so you'll have a long-lasting protection. Unlike CTL, okay, after an infection, CTL will rise, and then they're going to go down again. So you don't have very many CTL specific for a virus that you caught 3 years ago, if we're talking about an acute infection. Okay, one thing to remember is not all antiviral antibodies are neutralizing. So you can be seropositive by ELISA or Western blot, but that doesn't mean to say that these antibodies are actually having an effect on the virus. And this is why some vaccines or vaccine candidates don't work. So what are the applications of neutralizing antibodies in the clinic, for example? Well, serotherapy, right? You can transfer hyperimmune gamma globulin against different viruses, and they can be used for post-exposure prophylaxis. Babies who are born to mothers who have an active hepatitis B infection will get these hyperimmune globulins against HPV and then be vaccinated, and that can prevent these children from becoming infected. The same thing is if you get bitten by a bat and you're at risk for rabies infection, you'll probably get some hyperimmune globulin and then the anti-rabies vaccine. Respiratory syncytial virus, another one where this is an effective therapeutic approach. This is for babies who are born before term. They're very, very susceptible to severe respiratory syncytial virus infection. So this is the kind of proof of principle. And in fact, these therapies based on serum that can come from people are being replaced by monoclonal antibodies, either of single monoclonals or a cocktail of very, very active neutralizing monoclonals. And the reason these are more effective is because their neutralizing activity is better and you can deliver them in a smaller dose. So if you've got a young baby to inject, this is actually a problem for immune serum. If you can treat with a small volume of highly active monoclonal antibody, that's a big advantage, right? So for RSV, this is a product that's used for prophylaxis of RSV. This is a monoclonal antibody cocktail, 2 monoclonals, which is under development for rabies post-exposure prophylaxis. And then there's this one, the ZMapp, okay, which has been used to try and treat Ebola virus infection. So let's have a look at ZMapp a little bit and see how that was developed. In fact, there were 2 teams looking at this therapeutic approach, and they both developed cocktails of monoclonal antibodies independently. So these were published in 2012, these studies. And the kind of results that they got was that they were infecting, I believe this was macaques, with 1,000 TCID50 of Ebola virus intramuscularly. And then they gave the antibody cocktail, that's this one, 24 hours or 48 hours post-infection. So these are the non-treated animals. They all die within like 6 days. They're all dead. If you inject the monoclonal antibodies within 24 hours, they all survive. But even 1 day later, the survival rate goes down to 50%. So this is going to be very, very difficult to use this clinically, because you're never going to find someone who's just been infected yesterday because they're asymptomatic, right? Then they got together, in fact, these two teams, and thought, okay, we've both got these monoclonal antibodies. We've got 6 different monoclonal. Maybe we can define what is the best combination. So that's what the ZMAP is, okay? It's got 2 antibodies, or no, this antibody from this cocktail, and it's got 2 antibodies from the other cocktail. So they developed this combination and then tested whether it would be useful in post-exposure treatment of macaques with a symptomatic infection. So here's the protocol here. This was published in August this year. Once again, they're infecting the animals 1000 TCID50 on day 0. And then they're treating the animals either 3 days, 4 days, or 5 days after infection. Now, among these graphs, you try and have a little look at the black lines here. These are the placebo animals, or the non-treated animals. Okay, they're all symptomatic by 3 to 5 days. Temperature goes up. Everybody's got fever. Got a big rise in neutrophils that are circulating, and the platelets are going way down. So all of these animals are really ill by the time they're getting the antibody therapy. This is what happens. Okay, all of the non-treated animals, they're dead by like 7 or 8 days. All of the treatment groups, they all survive 100%. So this is, for me, this is really quite impressive, because you're giving your first dose here at 5 days. 3 days later, these animals are going to be dead if they don't get any treatment. And if you give these antibodies, then they seem to all survive. So this is pretty convincing preclinical data that this is going to work. The big problem, of course, is monoclonal antibodies are not cheap to make, and they are not easy to produce in very, very large amounts. So at the situation where we are now with this epidemic, there are going to be far too many people who need treatment compared to the amount of the therapeutic that can be produced. Okay, so neutralizing antibodies as therapeutics, that's one thing. They're also important in vaccination. Okay, so I think you can try and look at whether or not we have an effective vaccine. Depends pretty much on whether neutralizing antibodies are easy to induce against the virus. So for all these virus infections, there are either one serotype or a limited number of serotypes. So that means that it's been possible to develop a universal vaccine. By that I mean a vaccine that will be effective against all the strains of the virus that are in circulation worldwide, and it's going to be effective through time. Okay, so you don't need to change the vaccine. So this type of vaccine exists for all these viruses. And there are some viruses which pose problems. Okay, so rhinovirus and adenovirus, they have lots of different serotypes. So you can make a limited vaccine. So there are actually none that are used anymore. But for a while in the US, there was an adenovirus vaccine that was specific for 1 or 2 serotypes. It was used in the army and it was effective. But it was only effective against a very limited number of the viruses that were around. So these kind of limitations meant that they stopped using it. So for influenza, as you probably know, the antigenic makeup of the virus is variable over time. So this is what you call antigenic drift, and that means that the vaccine has got to be reformulated every 2 or 3 years. That's what I mean when I say it's limited. Okay, in this respect. And for HIV, we have another type of problem. Okay, so we don't have a universal vaccine and we don't have a limited vaccine either. So then the last part of the talk, like in the last 15 minutes, maybe 10 minutes, and we'll have a couple of questions. I'm going to look at what exactly is the neutralizing antibody problem in HIV infection, and are we going to be able to get around it one day? So this is an illustration of the neutralizing antibody problem in HIV infection. This is I love this experiment actually, or these observations. This is from a guy called Doug Richman. And what they did was they sampled the virus in an infected person at different times after infection. So they got virus at 0 months, as close after seroconversion as they could, or even before seroconversion. 3 months, 6 months, up to 2 years after infection. And they also got plasma from the same person at different time points. So for each combination, they tested the plasma that was present against all the different viruses that were circulating in that person. So at time 0, this is before seroconversion, right? The neutralizing titer is nothing, right? The higher this number is, the higher your neutralization is, okay? So anything less than 100 is negative, basically. So before seroconversion, you've got no specific antibodies. You don't neutralize any of these different viruses. At 3 months, you can neutralize the virus that initially infected you. And you've got some neutralization against the virus that's there. At 6 months, you can neutralize the virus that was present at 3 months and the virus that came in and infected you. But you've got no neutralizing titer against the virus that's actually there at this time point. And it goes on like this all the way through, like 12 months. You've got antibodies against all the previous viruses, but you've got nothing against the virus that's actually there at 12 months. So you can see that there's this kind of race, okay? The immune system is developing, selecting these neutralizing antibodies, but it's just selecting for escape mutations in the virus. And the virus always stays one step ahead. So that's why the humoral response in HIV doesn't manage to get rid of the HIV infection, okay? So people who are infected do develop neutralizing antibodies, but most of them have a very narrow spectrum of neutralization. And that leads to continuous escape of the virus through time. So where are these neutralizing antibodies binding to the virus, and do broadly neutralizing antibodies even exist, or is it impossible? So to think about this, let's just have a reminder of the steps in HIV-1, attachment and entry. So the functional form of the attachment glycoproteins is this trimer between 3 molecules of GP41, 3 molecules of GP120. GP120 is going to bind firstly to CD4. This interaction leads to the liberation of the V3 loop, which binds to the co-receptor, CCR5. And once CD4 and CCR5 have been bound, then the fusion domains in GP41 will induce fusion between the virus membrane and the host cell membrane. Okay? So these are 3 essential steps: CD4 binding, CCR5 binding, and deployment of the fusion domains. So neutralizing antibodies can inhibit any one of these steps if they fix the CD4 binding site, if they fix the CCR5 binding site, or if they fix the fusion domains. Now, what is the problem here? One of the problem is that, okay, a lot of the surface of GP120 is not involved in CD4 binding or V3 loop binding. So if you have antibodies binding around here, they're not going to be neutralizing at all. They're just going to be bouncing off the virus's armor. The V3 loop antibodies, they will be neutralizing, but the V here is for variable. This is a highly variable site. So these ones are going to have a very narrow neutralizing spectrum. Broadly neutralizing antibodies fall into maybe 2 or maybe 3 families, but I'm just going to talk about 2 of them. Ones that fix the CD4 binding site, that's here, and broadly neutralizing antibodies that bind to the fusion domain. So once you know this, then there's been a very large effort. You know, firstly, it took years actually to try and screen hundreds of patients, try and pull out B lymphocyte clones that express these broadly neutralizing antibodies, because most people don't develop this kind of antibody. And then find out where they bind, and then the next step is, can we get a vaccine that will induce the expression of this kind of antibody? Oh, okay, before I do that so this is it. Can we induce broadly neutralizing antibodies? Now we know what they are, okay? Can we actually induce these things, right? So let's have some information from this HIV vaccine trial, RV144. This was a phase 3 trial. It enrolled 16,000 volunteers, and about 8,000 people got placebo, 8,000 people got the vaccine. The vaccine was a recombinant canary pox, which was boosted by monomeric GP120. So you might expect that you're getting some kind of antibody response against GP120 here. And this is a very remarkable study because it's the only HIV vaccine trial that showed evidence of some kind of protection. So it was calculated that it was about 30% protection amongst the people who received vaccine. That's encouraging. But when you look, in fact, there's no evidence of induction of broadly neutralizing antibodies in this trial. So there is some protective effect, but it's not due to the broadly neutralizing antibodies. There's still some debate about what really was the mechanism of protection here. Well, one thing we can say, though, that these immunogens don't seem to be sufficient on their own to induce broadly neutralizing antibodies. So what are the strategies that are being developed? Well, one of them is to try and redesign the structure of GP120 and form trimers or multimers that render this CD4 binding site much more immunogenic. Okay, so there's a fantastic piece of work that was published in Science last year. It's really great. So maybe this will be something that can work. And another approach is to try and induce antibodies directed against the fusion domains, directed against the GP41 epitopes. And this is Morgane Bumsel in Cochin who's been doing this. So in 2011, she showed that this was possible in macaques, and phase 1 has just been published this year. What is interesting about this is that she was very careful to try and target induction of antibodies at mucosal sites. So there's a prime that's intramuscular, and a boost is intranasal. So you're trying to get the secretion of mucosal antibody. You're trying to get IgA, IgG at the mucosa. This is the site of the infection. So these two are both, very quite promising strategies. So maybe one day we actually will get an effective HIV vaccine. Okay, I think I'm more or less finished. So let's have a little question about CTL. So if you all go back onto, oh no, I'll quit this. You all go back to your devices, right, and get back onto here. I'm going to pause, right? Okay. So, and I'll try and give you a new question. Okay, so you should get a new question now. You got it? Oh, it should be started again. Oh, yeah, okay, I did it wrong. So you will need a new session key. 16747. It's here. In fact, it's possible to use the same key, but you have to do it. You have to do the steps exactly right. I'm not an experienced enough user to get it right every time. Okay, everybody got this key? 16747? So you should get some questions about CTL. Oh, it's good, this thing gives me a break. Okay, I've got 7 responses. I'll try and wait till we have 8 or 9. We got 9 last time, right? So let's try for 9. Yeah, okay, thank you. So let's see what you got. Okay, CTL are less important than antibodies in the resolution of infection. Okay, you're right. Nobody said yes to that. You are all correct. They are very important in the resolution of an infection. Okay, question 2. Cells that are infected by a virus that inhibits apoptosis cannot be killed by CTL. 2 people said yes. That's wrong, actually. Okay, so CTL effectors, granzymes that are secreted into infected cells, they can kill a cell even if it's resisting apoptosis. Okay, these granzymes are going to have a direct effect on the cell. So next question is 3. DNA viruses can't escape from the CTL response because their mutation rate is too low. 3 people said yes. You are wrong. They can escape because they express these proteins that inhibit the different steps in the presentation of CTL epitopes. Remember, you know, the CMV proteins, Epstein-Barr virus, they express proteins that inhibit presentation of CTL epitopes. So 3 people fell into the trap here. 6. The initiation of a CTL response is dependent on the activation of the innate response. Yes, that's correct. Okay? So 7 people got this one right. If you don't have TLR activation on dendritic cells, you can't start your adaptive immune response. So you won't get a CTL response unless you have an innate response first. Now then, NK cells recognize viral peptides on the surface of the infected cells on HLA class 1 molecules. 6 people said yes. Mostly that's wrong. That's wrong, right? They're recognizing the absence of HLA class 1 and they're recognizing the presence of these activator receptors. They're recognizing the presence of MIC-A, MIC-B that are going to bind to NKG2D. So generally speaking, most people got this one wrong, I think. Okay. That being said, okay, there is some indication that the signal that comes through, the negative signal that comes through KIR receptors does depend on the peptides that are bound. So if you have an if you have an infected cell, the peptides that are displayed on the HLA class 1 are not going to be the same as in a non-infected cell, and this will affect the negative signal that is given to through the KIR receptors. So generally speaking, I would say that this answer is wrong, but if you explained exactly what you meant, you could get away with it. Okay. Right, thank you for playing the game here. So I'm just going to go through the summary and then that will be finished. Okay, so the main points once again. How do you know that how does a cell know it's infected? It's basically these receptors that detect viral nucleic acids. When viruses are detected, the 3 consequences. Interferon, okay, it's going to protect the surrounding cells. Programmed cell death. The infected cell will be killed. Often this depends on these nucleic acid receptors. 3rd one, dendritic cell maturation. That's essential for starting the adaptive response. What do you do if you're infected? Well, secrete interferon, and this will induce restriction factors that will help the surrounding cells to resist viral infection. Cellular response, okay, the main role of NK and CTL is to detect and kill infected cells. They have okay, you know this, you just answered the question, so I don't need to say this. Doesn't seem to really work for durable protection induced by vaccination. Okay, works in animal models, doesn't seem to work in human cases. One of the differences might be that in the animal model, you choose when you perform the challenge. So you can choose to put your virus and the infection in, you know, quite recently after the vaccine, so the CTL is still there. But in real life, after a vaccination, you never know when the person is going to be infected. So that might be why it works in an animal model. It does not seem to work in real life. Humoral infection and humoral response and vaccination. Okay, make sure you know what neutralizing antibodies are, that they are used as part of post-exposure prophylaxis for some viral infections, and the effective vaccines that we have seem to work because they can induce neutralizing antibodies. And where we don't have an effective universal vaccine, there's some problem to do with, how can we induce a broadly neutralizing response? So that's what I think, anyway, the future challenges in vaccinology. So I'll leave it there. Thank you for playing along with my little game on M-clickers. If you have any questions, I will be happy to completely ignore them and leave you in ignorance.